Antibiotic resistance is driven by various genetic mechanisms, which can be broadly categorized into three types: intrinsic resistance, acquired resistance, and cross-resistance. Intrinsic resistance refers to a bacterium's natural ability to resist certain antibiotics without prior exposure due to its physiological traits or structural genes. For instance, Mycoplasma bacteria which lack a cell wall are naturally resistant to penicillin, an antibiotic that targets bacterial cell walls. Natural selection plays a key role in the spread of antibiotic resistance, as bacteria that survive antibiotic exposure reproduce while susceptible strains die off. For instance, methicillin was once effective against Staphylococcus aureus, but strains carrying the MECA gene have become dominant, rendering methicillin less effective. Acquired resistance arises through resistant mutations or horizontal gene transfer. Resistant mutations can happen spontaneously due to errors in DNA replication or can be induced by environmental stresses such as antibiotic exposure. Horizontal gene transfer involves three mechanisms: conjugation, transformation, and transduction. Conjugation involves the transfer of plasmid DNA, small circular DNA molecules, from one bacterium, the donor, to another, the recipient, through direct contact via a pilus, thereby spreading resistance. Transformation occurs when a bacterium takes up extracellular DNA from its environment, often from live cells. This DNA containing antibiotic resistance genes can be either chromosomal DNA, which integrates into the bacterium's genome through homologous recombination, or it can be plasmid DNA, which self-replicates independently within the cell, enabling the expression of resistance traits. Transduction occurs when bacteriophages, viruses that infect bacteria, mediate the transfer of chromosomal or plasmid DNA from the donor bacterium to the recipient, spreading antibiotic-resistant genes without direct contact. Cross-resistance happens when bacteria develop resistance to multiple antibiotics in the same class due to shared mechanisms of action. For example, Staphylococcus aureus resists the MLSB group of antibiotics, which, despite having different chemical structures, all inhibit protein synthesis by binding to the 50S ribosomal subunit. This resistance is mediated by genes, which modify the ribosomal target site, reducing the binding affinity of MLSB antibiotics. In summary, these genetic mechanisms demonstrate how bacteria evolve defenses against antibiotics, contributing significantly to the growing challenge of antibiotic resistance.